The topic for today's OICR Monday session is the Hippo Pathway in Cancer and Fibrosis. The format for today's session will be an introduction by the moderator, followed by the three short presentations from our speakers. Each speaker represents an OICR research theme. Now I would like to introduce today's moderator, Dr. Liliana Attisano, who will guide us through the event today. Dr. Attisano obtained her PhD at the University of Toronto and was a postdoctoral fellow at the Sloan Kettering Cancer Institute in New York City. She's currently a full professor in the Department of Biochemistry at the Donnelly Center at the University of Toronto and is director of the Applied Organoid Core Facility that she established in 2019. The Attisano Lab is focused on studying the molecular events that underlie signaling cascades such as TGF-beta, Wnt, and Hippo, and elucidating how cells interpret contextual cues to control complex biological responses. Our labs use cell, organoid, and mouse models to understand how disruptions in these pathways contribute to human diseases, including cancer and CNS diseases. Her work, which is cited over 32,000 times, has provided seminal observations on molecular aspects, development functions, and how mutations in signaling pathways contribute to disease. Liliana, thank you so much for agreeing to moderate today's session. I'll turn it over to you now.
Thank you so much. It's my pleasure to talk about one of my favorite pathways today and hear about the exciting talks we have planned. As you all know, today we're going to be focusing on the Hippo pathway in cancer and fibrosis. And I just wanted to highlight the fact that the Hippo pathway is a lot different than most of the signaling pathways you may be familiar with. And I just want to start by introducing that to you. So basically, at the core of the pathway is a pair of kinases called MST and LATs, and we call this the Hippo kinase cassette. And its role is to regulate the activity of YAP and TAZ. The kinase cassette and the transcriptional regulators are key regulators of tissue and organ growth. And a lot of studies in flies and animals have taught us the core features of the pathway. So if you inhibit the Hippo kinase cassette, what happens is you get this organ overgrowth. And that's because there's excessive signaling by these transcriptional regulators, YAP and TAZ. And so this was one of the earliest discoveries of the pathway, and they called it Hippo because I guess the flies look a little bit like a hippo with this overgrowth. And so if you knock out the cassette, you get overgrowth. And if you overexpress the transcriptional regulators, you similarly get this overgrowth. The pathway is conserved in higher organisms. So for example, in mouse, if you knock out the kinase cassette, you get liver overgrowth. And similarly, if you overexpress the transcriptional regulator, same thing happens. You get a very large liver. Now, as you can imagine, with this overgrowth phenotype, a short-term increase causes organ overgrowth, but upon prolonged activation of the pathway, it eventually leads to tumors. And so one of the interests of Hippo pathway is trying to regulate it as a therapeutic for cancer. So just to step back a bit and give you a little bit more details. Here's the kinase cassette that includes MST and LATs, and there's also these adapter proteins that are required to act within the complex. And basically, MST will phosphorylate LATs, which gets activated. LATs will then phosphorylate the transcriptional regulators, YAP and TAZ, and keep it in the cytoplasm. So when the pathway is on, transcription is off. So think about it opposite to your normal pathways. When the pathway is off, YAP-TAZ will go into the nucleus, cooperate with DNA binding transcription factors called TEADs, and thereby activate transcription. And the fly pathway is very similar. So again, unlike other signaling pathways, there's not a single ligand receptor intracellular cascade that leads to the regulation. Rather, the Hippo pathway is able to sense the environment and respond to a huge plethora of cues. So for example, cell-cell contact or polarization of cells will activate the pathway. Things like GPCRs are known to regulate the pathway, and also mechanical cues, stiffness of the matrix, flexibility, that kind of thing. So all of these different signals, and these are just a few shown here, basically regulate the activity of MST and LATs and either phosphorylate or not YAP and TAZ to regulate transcription. So there's no obvious route if you wanted to block the pathway upstream of the kinase cassette. And one of the easiest ways to tell what's going on with the pathway is just look for localization. And I just picked an example. Here, these are cells, mammary epithelial cells placed at low density, and we're looking at the localization of YAP and TAZ. And here at low density, YAP is in the nucleus. And at higher density, when there's strong cell contacts or when cells become polarized, it moves into the cytoplasm. And so that's kind of a characteristic feature of the status of the pathway. And the last thing I just wanted to point out is that Hippo pathway does not act in isolation. There's numerous other signaling pathways that it can interact with. I picked two that were related to some of my own research, but there are certainly others. So the status of Hippo can regulate TGF-beta signaling or Wnt signaling. Thus, manipulating the core Hippo pathway can have impacts on the other pathways and vice versa. So something to keep in mind when studying the pathway. And that's all I wanted to do to introduce you to the basic features of the pathway. And we'll hear more about it in the next set of talks. So our first speaker is Catherine Song. Catherine is actually a PhD candidate in my laboratory. She comes in through the Department of Medical Biophysics. She completed her undergrad degree here at U of T, majoring in biochemistry and physiology. And her research project is focused on identifying NUAK regulators. And Catherine's going to tell us a bit about the role of NUAKs in cancer and fibrosis. Thank you, Catherine. Go ahead.
Thank you, Liliana, for the introduction. Hello, everyone. It's a great pleasure to participate in the OICR Monday series and introduce to you some of the work from our lab regarding the Hippo pathway. I'm Catherine from Attisano lab, and today I'll focus on the identification and role of NUAKs in the Hippo pathway and their implications in cancer and fibrosis. So you just see it a minute ago, so I'll go through this very quickly. Essentially, the Hippo pathway is composed of a kinase cascade and functions as a key regulator of organ growth and tissue size. So when Hippo is on in response to these upstream regulators, MST and LATs in complex with their adapter proteins, phosphorylate YAP and TAZ that promote cytoplasmic retention and sequential degradation of YAP and TAZ. When Hippo is off, in their unphosphorylated form, YAP and TAZ function by translocating into the nucleus and interacting with several transcriptional factors, such as TEAD, to regulate the expression of genes involved in cell proliferation, survival, transformation, and migration. When the pathway is dysregulated, abnormal transcription will ultimately lead to cancer and fibrosis. Given its widespread roles in normal development and homeostasis, it's not surprising that the Hippo pathway has now been linked to diverse human diseases, and many gene mutations of the Hippo pathway components have been identified in cancers and non-cancer diseases. As a matter of fact, the Cancer Genomic Atlas identified the Hippo pathway as one of the 8 major signaling pathways commonly altered in human cancers. In comparison to mutations of upstream tumor suppressors of the Hippo pathway, activation of YAP and TAZ is even more prevalent in human cancers. For instance, in breast cancer, high YAP and TAZ activities correlate with high histological grade, enrichment of stem cell signatures, metastasis, and poor outcome. And on the other hand, abnormal YAP and TAZ activity is also associated to fibrosis, which is usually caused by disease-related injury that triggers a cascade of defective wound healing responses, resulting in excessive accumulation of extracellular matrix. Fibrosis is a pathology feature of disease in multiple major organs such as liver, lung, and kidneys. The role of YAP and TAZ in fibrosis in various organs has been extensively studied, such as in kidney fibrosis. Elevated YAP levels were reported in renal fibroblasts during interstitial fibrosis in mice, leading to increased myofibroblast formation and ECM production. And similarly, activation of YAP in renal proximal tubule epithelial cells also drives renal interstitial fibrogenesis. With the importance of Hippo pathway in cancer and fibrosis in mind, our lab has been working on identifying upstream regulators of YAP and TAZ, which will provide more druggable targets in this pathway. To do that, we have performed a siRNA kinome screen using an imaging-based YAP and TAZ subcellular localization assay as readout and comparing with the TEAD luciferase reporter assay results. And this identified NUAK2, which is an AMPK family member and closely related to its paralog NUAK1. NUAK2 is regulated by various stress stimuli and has implications in cell survival, migration, and invasion. NUAK2 is also frequently amplified in many cancers, suggesting its potential roles in tumor development. And in the case of breast cancer, NUAK2 has increased expression in various subtypes, and in colorectal cancer, its paralog NUAK1 is also overexpressed in the subtype CMS4, which has mesenchymal characteristics and is associated with worse prognosis after relapse and overall survival. So to investigate the effect of NUAKs on tumor growth, different groups have developed mouse models with distinct tumor types. Our lab found that CRISPR knockout of NUAK2 blocks tumor growth in an orthotopic memory model. As shown here, the tumor growth is significantly delayed and epithelial mesenchymal transition is suppressed, as indicated by the Vimentin staining. And similarly, the Camargo group developed a YAP-induced liver tumor model, and they also found that knockout of NUAK2 or chemical inhibition significantly reduced liver overgrowth and hepatocyte proliferation. Furthermore, the Murphy lab looks at NUAK1 knockout in a colorectal cancer model, where they induce sporadic colorectal tumors with the loss of APC and mutation of KRAS, and found that the deletion of NUAK1 profoundly suppressed both the number and size of individual tumors in the colon. All these data indicate that NUAKs play important roles in tumorigenesis. Therefore, we started looking into the mechanism of NUAKs promoting cancer cell proliferation. We first verified our kinome screen results in breast cancer cells, and consistent with our screening data, knocking down NUAK2 using siRNA, genetic ablation by CRISPR, or chemical inhibition all promoted cytoplasmic localization of YAP and TAZ and reduced expression of YAP and TAZ downstream target genes, such as NKRD1 and CTGF. The same effect is also validated in high-grade bladder cancer cells, and we observed a significant cell growth suppression. So these indicate that the loss or inhibition of NUAK can restore the Hippo pathway and can decrease YAP and TAZ activity. We next looked at the effect of NUAK on YAP localization by live cell imaging of cells expressing wild-type YAP or constitutively active YAP. The mutant contains 5 serine to alanine mutations, all of which are at LAT's targeted sites, rendering them resistant to cytoplasmic sequestration and degradation. As you can see in the movie here, by adding the NUAK inhibitor, YAP rapidly translocated into the cytoplasm. as shown here. And while on the other hand, with the mutated YAP, all YAP remains nuclear even after the addition of the NUAK inhibitor WZ4003, suggesting that inhibition of NUAK promotes nuclear export of YAP, which is dependent on YAP phosphorylation by LATs. And next, we sought to answer the question whether NUAK regulates Hippo pathway activity through LATs. We confirmed the interaction of endogenous LATs with flag-tagged wild-type and kinase NUAK. We then examined whether LATs is required for NUAK to function on YAP and TAZ. And as expected, loss of LATs1 and 2 promoted nuclear localization of YAP and TAZ and enhanced expression of YAP and TAZ target genes. Importantly, the loss of LATs also reversed the inhibition of YAP and TAZ nuclear accumulation and target gene expression, which is caused by SI-NUAK2. And together with other experiments, we identified NUAK2 as a positive regulator of YAP and TAZ activity and a positive feed-forward loop in the Hippo pathway, where YAP and TAZ induce expression of NUAK2, which in turn acts to inhibit LATs to enforce a strong YAP and TAZ transcriptional program, promoting cell proliferation and tumor growth. Our ongoing work, including identifying NUAK2 regulators in the Hippo signaling pathway in order to provide more understanding of the role of NUAK in cancer contexts. Now, on the fibrosis side, in collaboration with the Yuan lab and Rana lab, we identified NUAK1's association with renal fibrosis and declining kidney function in human kidney transplant patients. By plotting the correlation coefficients for each transcript in a 2D matrix, we revealed a clear association with fibrosis and kidney function. Among the putative disease drivers, as indicated in the red dashed box, we found NUAK1 along with other TGF-beta family ligands such as TGF-beta 2 and activin A. To briefly introduce the TGF-beta family, they are the primary factor that drives fibrosis in almost all forms of chronic kidney diseases. The induction of TGF-beta signaling pathway by its ligand leads to the phosphorylation and activation of SMADs, which then translocate into the nucleus, bind directly to gene promoters to induce transcription of pro-fibrotic molecules, such as alpha smooth muscle actin, collagens, and fibronectin, which then induce myofibroblast activation and extracellular matrix deposition. And we then analyzed NUAK1 expression profile, and its expression was strongly correlated with the fibroblast TGF-beta signature. Given these findings, we next examined the functional relevance of NUAK1 in fibrosis. We know that both YAP and TAZ and TGF-beta are critical regulators of fibrosis in many different tissues, and growing evidence suggests that TGF-beta signaling is closely linked with YAP and TAZ activity. Therefore, we hypothesized that NUAK1 might serve as a critical link between these two pro-fibrotic pathways. And to test this hypothesis, we induced TGF-beta signaling in fibroblast and then treat the cells with a NUAK inhibitor. In the TGF-beta stimulation group, YAP translocates into the nucleus, while adding the inhibitor completely reverses this effect, suggesting that NUAK inhibition decreases TGF-beta-induced YAP activation. Likewise, the expression of downstream YAP target genes and fibrosis-associated genes were also reduced after NUAK1 inhibition. To confirm these results, we next silenced NUAK1 in fibroblasts using siRNA and then co-transfected with wild-type or kinase mutant of NUAK1. As shown in the results here, NUAK1 knockdown has similar effects as chemical inhibition we just saw, attenuating the ability of TGF-beta to induce the expression of YAP target genes and pro-fibrotic genes. And this effect was rescued by the wild-type but not kinase NUAK1, indicating that TGF-beta-induced gene expression requires NUAK1 kinase activity. And together with other experiments, we observed a positive feed-forward loop in the fibrosis context as well, where NUAK1 is induced by TGF-beta, YAP, and TAZ, but also acts back as a critical regulator of YAP activation and SMAT signaling, which ultimately drives fibrogenesis. To better understand the role that NUAK1 plays in regulating fibrosis in vivo, we generated a fibroblast-specific NUAK1 deficient mouse model where kidney fibrosis is induced with unilateral ureteral obstruction. Consistent with our in vitro findings, fibroblast NUAK1 deficiency greatly attenuated UUO-induced kidney fibrosis, which is marked by the staining of fibrillar collagen. And lastly, we treated mice with the NUAK inhibitor after UUO and observed the same attenuation effect of kidney fibrosis as in the genetic ablation experiment, providing an exciting opportunity of NUAK inhibition as an anti-fibrotic therapeutic strategy. So to summarize, dysregulated Hippo pathway is known to be associated with tumorigenesis and fibrosis. NUAK1 and NUAK2, two AMPK family members, have critical roles in the pathway and are overexpressed in various cancers. We showed that NUAK2 and YAP and TAZ form a positive feed-forward loop that promotes tumor growth in breast cancer model, and a similar pro-fibrotic positive feedback loop also exists between NUAK1 and YAP and TAZ that enables fibrogenesis in kidney. Altogether, these findings provide us insights into the roles of NUAKs in the Hippo pathway and offer new druggable targets in treating cancer and fibrosis. And with all that, I'd like to thank all my lab mates in the Attisano lab and our collaborators for their tremendous help in the NUAK projects. I'm happy to take any questions in the discussion period afterwards. Thank you.
Okay, thank you very much, Catherine. And it's my pleasure to introduce the next speaker, Dr. Methvin Isaac, who's a senior scientific advisor in the drug discovery program at OICR. Methvin has over 20 years experience in identifying drug candidates in a variety of different therapeutic areas, including CNS, GI, and oncology. Right now, he's the medicinal chemistry project leader of 2 OICR collaborative projects, including one on the NUAKs. And prior to joining OICR, his efforts have contributed to the discovery of many new chemical entities, including 6 drug candidates. And he's an inventor on a remarkable number of 75 issued and published patents. Thank you very much for agreeing to talk, and we look forward to your presentation today, Methvin.
Okay, thank you, Liliana. Hi, everyone. So thanks again, Liliana and Jay, for inviting me to speak. So you just heard a very nice overview of a lot of the background biology for a very intriguing pathway and revealing some exciting potential targets for therapy. And so being a medicinal chemist and, from a drug discovery background, we actually now look at some of that biology and start thinking about potential ways to develop small molecules to drug this target. And so what's exciting about this project is, this is a particular project that really involves collaborative effort. So you're kind of, relying on all of that brain power in the local environment in Toronto. So luckily for us, for our drug discovery team, we were able to collaborate with Liliana and her team based on her discovery of the target. And in close proximity, we have Frank Sekiri, who is a crystallographer, have a crystallized AMPK-related structure. So NUAK kinases are very tough to crystallize, but a lot of homology studies have been done based on crystallization of related family members. And Dr. Anna, he has quite a vast experience in some of the TGF-beta related models and some models we can potentially use in this project. So, I'm not going to belabor the point that you just heard, that the Hippo pathway is very central to a lot of cancer types. I mean, it's essential in early development of organs, but, tend to get silenced, and then cancer somehow hijack this pathway for their benefit later on. But luckily, with that discovery that you just heard from Attisano's group, it really presents an opportunity to really target NUAKs to inhibit this pathway and actually inhibit cancer growth. But a lot of therapeutic strategy currently going after the Hippo pathway involves, disruption of some of the components, the nuclear components, that is the TEADs, disrupting the interaction of YAP and ARTAS with TEADs, which present a sort of a protein-protein interaction strategy. Again, you're drugging a transcription factor. So those were previously known to be notorious, difficult targets to drug, transcription factors and protein-protein interaction. So what we find really appealing about the NUAK strategy from Liliana is that it presents, a very druggable target, kinases, which are well established and have clinical precedence with a number of drugs going into the clinic. So we really found that appealing. So we believe that the NUAK strategy allows a probably easier druggable strategy towards, modulating the Hippo pathway relative to some of the TEAD strategy which have been out there. So just to give you some drug discovery background, now the current thesis for the project, as you just heard, is we believe that if you inhibit the NUAKs, preferentially NUAK2, then you'll be able to modulate the Hippo acid and impact the pathway. And what's also exciting about this strategy too is, a lot of times when people identify targets, then the obvious next step is to look at clinical biomarkers, which can sometimes be an uphill battle. But we are blessed by the fact that such strategy in the Hippo pathway already has established clinical biomarkers. And Liliana just mentioned a couple of them which you can actually use to really interrogate whether or not the transcription pathway is on or off. Things like, CYR61, CTGF, and ANCARD1. Those are good downstream targets you can actually track as clinical biomarkers with regards to therapy. So now we have seen a lot of the biology that went into genetic ablation and really demonstrating that these NUAK targets are really essential for driving some of the growth of some of these tumors. And so by blocking them either with CRISPR, shRNA, and a weaker NUAK inhibitor, you get nice blocking of the cancer-promoting effects, and you also see that being made evident by knocking down the target genes associated with YAP and TAS. So with that genetic background, as discovery chemists, we always want to know if you can phenocopy that, genetic ablation with small molecule inhibitors. So that became a task for us. And so we established a cascade, just like any drug discovery program, where the hope was to design molecules. We can use some of that design process using the NUAK homology model. Like I mentioned to you, NUAK, there's no crystal structure of NUAK. It's a very tough kinase to crystallize, but you have closely related family members, which provide, certain, binding motifs that we can use for homology modeling to design compounds. The plan is to then screen these compounds at NUAKs 1 and 2. And we do that at a CRO called Eurofins. And then internally, we develop a phospho-YAP assay that we look at phosphorylation of the serine 127 residue of YAP. And at the same time, like Liliana showed you, we can physically look at YAP localization events, and they correlate very nicely. The phospho-YAP assay and the YAP localization assay, they're very, very tight correlation between those two. After we identify compounds that we know inhibits the target and subsequently demonstrate modulation of the Hippo signaling through phosphorylation of YAP, then we start looking at some of the ADME properties, identify compounds with good PK, and then hopefully we can now take, good molecules into in vivo studies, whether it's fibrosis model or the in vivo model. So some of that I'll show you in a minute. What's exciting about this project, you'll see, is it's funny how serendipity plays out a lot of times in science. So Liliana was doing her, remarkable discovery of NUAK as a target. At the same time, when, our medchem department was established, one of the strategies we had, since a lot of us had come from industries and a lot of us had medchem backgrounds, we always wanted to create some libraries. So we create some proprietary scaffolds. Some of them were related to kinases, and we made a whole bunch of compounds to see if we can design novel scaffolds. And then we took some of these scaffolds and then we send them to Eurofins, and we sort of decide, let's see what sort of kinase targets they're going to hit. So what we call a chemocentric strategy. So we send these compounds out and we screen across the entire kinome at Eurofins. And lo and behold, a couple of compounds came back hitting NUAK because we were looking for new, kinases that nobody had actually drugged it with these new scaffold, with the hope that these new scaffold immediately provide an avenue towards IP. So when this screening was done, one of the hits that came back from our screen was this compound, OICR 1622. We realized this compound was hitting NUAK, and then we ran into Liliana, and then she said, "Hey guys, I'm looking for some NUAK inhibitors. Do you guys have any?" And it was just laughable that right away you see the opportunity to just collaborate. And so it was really, really a good opportunity for us to now, mesh a lot of the chemistry power that we had internally with some of the nice biology that you just see genetically. And so with this particular hit, 16422, we knew it hit NUAKs from the chemocentric screen. We knew it was novel because that was our goal, designing novel cores and stuff. It was potent, it was selective, but it had no good oral properties. And so that was one of the tasks for us at the outset, is we really needed to identify an orally bioavailable compound. But when we started digging into the literature for NUAK ligands, we realized that there was little to no NUAK2 inhibitors in the literature at the time. And there was a number of NUAK1 inhibitors, but a lot of them weren't really active at NUAK2. So we realized that right at the outset, we had a NUAK inhibitor. That was the most potent NUAK inhibitor we had identified, that was reported at the time. So it really, really seemed like a good opportunity. Now, all we had to do is keep an eye out for the Aurora activity, just because of the nature of the scaffold that we knew from a lot of our kinase knowledge. And so with that said, we established a target product profile. What is it that we desire? We wanted to see compounds that hit NUAKs. We wanted to see good cellular activity in terms of YAP localization and phospho-YAP, get a little hint of antiproliferative effects, run broad screening across the kinome, make sure we get reasonable selectivity, and you'll see some of that. And then we have some of the ADME properties. And what's key again, like I told you, the early hit compound never had much exposure. We really wanted compounds that were orally bioavailable, with the hope that we can put these compounds in vivo models of fibrosis and tumors. And I'll show you some of that data. So just to give you sort of a sense of some of this evolution here. So in addition to 16422, another hit that we got from this screen, which was a known compound that was in the literature, and Liliana had used this in her early screens, which was a WZ4003. That compound was weakly active at the NUAKs, but it was also weakly active in the phospho-YAP assay. So Liliana had to use it at high concentrations to see some of her efficacy, close to 10 micromolar. And that was evident from the phospho-YAP activity. But bottom line, that commercial compound, WZ compound, it was not novel and it was not very selective. So with 16422, you could see we had very good potency, and now we start seeing activity in the PAP assay around 500 nanomolar. We knew it was novel, we knew it was selective, but it was not orally bioavailable. But then saw evolution from the med chemist quickly identified 19451, which we consider as our early tool compound. It maintained good activity at NUAK 1 and 2. It was novel. It was moderately selective. It got about 10 to 30-fold selectivity over some closely related kinases. But the key for us was, as a breakthrough, was the oral bioavailability, as I will show you in the next couple of slides. So we had these dual compounds, which we could hope would now help us to demonstrate whether we can phenocopy some of that genetic biology. So here's a kinase profiling of 19451. What we do, so Eurofins have a diversity panel, there's almost 430 something kinases in the kinome. So what Eurofin does is they took representative kinase from all of the different trees in the branch, and they offer a panel to screen just to get an early sneak peek of potential selectivity. If you have a dirty compound, then you'll inhibit a lot of these kinases. So at a first glance, we were quite happy to see that 19451 selectivity profile wasn't that bad. And so this is percent activity. So, few kinases here were inhibited. at almost 300 nanomolar. So we decided this might be good enough to do some early interrogation of the biology. And like I said, the PK for this compound was very, very good in mouse. You can see here. We also run the PK in rat. We were getting almost 90% oral bioavailability. We were getting, micromolar exposure after dosing this compound at like 10 mg per kg. So this was quite exciting for us. We had two compounds that we can actually put in vivo. The other thing was to take 19451 and the lead compound 16422 and just quickly put them into some of the models where it is believed that these cancer cells are really YAP-driven. So there are 3 examples here where we have the head and neck cancer line called PDX005. We tested the lead compound 422 in an antiproliferative assay, 5-day assay, and we saw about 200 nanomolar activity for both that and 19451. In the breast cancer line, with a similar activity across the board, and then we pick a colon line because these are 3 tumor types where the Hippo pathway is highly deregulated, and so developing inhibitors of the Hippo pathway would actually be important therapeutics going after some of these cancer types. What's also exciting too is, there is an APC mouse model. And like I told you, Jeff Rana is an expert in developing these tumor models. And Liliana also had access to some of these mouse lines. So she was able to do or generate organoids and look at the inhibition of some of these compounds in the organoids. Here she tested 1642 and showed that you get nice growth inhibition of 1642 in these organoid models, presenting the potential for these compounds in cancer models. The next thing we did, obviously, was to test it also in the fibrosis model. Just like you see, Christine presented some of the nice genetic work that was done in mouse fibrosis. And so, again, the same UOO model that she talks about, we evaluated, the 1642, which we know wasn't orally bioavailable. So we dosed that IP at 30 mg per kg twice a day. And then we dosed the oral compound 19451 at 2 doses orally. And at that time, we ended up using nintedanib. Now, this is an approved drug for IPF, lung fibrosis, and it had been tested in the UOO model. So we thought that this was a good clinical benchmark, quite a standard of care in fibrosis. And so we use that as a positive control in our model. And so you could see, so nintedanib, which is 1143, dosed at 50 mg per kg, is shown here. And then our compounds, you could see, nicely block the accumulation of collagen. Okay, when dosed at the 30 mg per kg IP or even orally in this model as shown in the graph in the middle. So we're excited to see that we were getting efficacy in collagen deposition. And then you could correlate that to some of the target genes in fibrosis, which is collagen 101. So when we do the target gene analysis, we got nice correlation between the ability to block collagen deposition and the correlation between target gene analysis like COL 101. So it's a nice efficacy in the in vivo model. We then tested, this was just before the COVID, we then tested quickly some of these compounds, 1642 and 19451, in the head and neck cancer model that was available from Dr. Shramek's lab. And so, we quickly saw some hint of efficacy from 1642 and 19451. But because these are proof of concept study, what was really important for us is to actually look at, are we on target in these tumors? So some of these tumors were isolated and YAP staining was performed. And what's exciting to see is that compared to vehicle, you could see all of these tumors had a significantly diminished level of YAP, suggesting that we're actually on target blocking the Hippo pathway. Similarly, Jeff Reiner had also developed this orthotopic model, MDA231. And so he looked more at the Kaplan-Meier analysis using the same 60 mg per kg. We got very, very nice efficacy at 60 mg per kg. He also did staining with YAP and TAZ at 60 mg, and we see nice efficacy in terms of the ability to block YAP staining in some of these tumors. But what was even more intriguing is you know, the ability of breast cancers, orthotopic breast cancers, to metastasize to the liver. And it looks like the 60 mg dose blocked that ability of cancers, of the tumors, to metastasize to the liver. As shown here in the vehicle control, you see all these different spots. That is the breast cancer metastasizing to the liver. That was totally blocked with the 60 mg dose. So that was very, very exciting in terms of, you know, a pharmacological strategy to block, you know, these triple negative type of breast cancer, and at the same time, you know, mitigating metastasis. So yeah, so you got a flavor of the genetic aspects. Now we are able to now find compounds that might actually be showing up some of the pharmacological aspect, presenting a very attractive strategy to drug the Hippo pathway. So to sum up, we believe that, you know, we have generated some compelling biology, you know, that characterizes NUAK small molecule inhibitors as Hippo pathway modulators. We identify potent, selective, and orally bioavailable inhibitors, and demonstrated efficacy in some in vitro cancer models, in vivo models, and some fibrosis models. Our lead compound attenuated fibrosis in the UOO model. And we also show that it inhibits some YAP-driven tumors. We have filed IP on some of these earlier, you know, lead scaffolds, and we believe that this presents an opportunity to develop first-in-class NUAK inhibitors. So what are some of the key next step that's left is we want to continue to do lead optimization, and identify preclinical candidates, and hope we can do comparisons between TEADs to show if there is any sort of differentiation between directly inhibiting the TEAD-YAP interaction or going after NUAKs, preventing the entry of YAP into the nucleus. We want to explore these lead compounds into additional patient-derived organoid models, colorectal organoid models that we have access to. We also want to look at some of the other cancer indications, head and neck, triple negative breast cancers, and keep an eye on kidney, lung, and liver fibrosis. And we're hoping, in short, that we'll be able to identify a preclinical candidate and start to launch IND-enabling studies. So I think that's about it.
Thanks a lot, Methvin. So I'm here now to introduce our third and final speaker of the session. It's my pleasure to introduce Dr. Tracy Tang. Tracy has over 20 years experience in cancer research and comes to us currently as the Vice President, Head of Biology and Translational Medicine at Vivace Therapeutics, a company focused on cancer drugs targeting the Hippo pathway. She has extensive prior experience in drug development, previously having been at Oncomed Pharmaceuticals, working on the anti-Notch and anti-Wnt clinical trials. And prior to that, she was at Genentech and was a major contributor to the development of the hedgehog pathway inhibitor, which I'm sure most of you are aware of. She earned her PhD at MIT, and prior to that, a Bachelor of Science focused on biochemistry from the University of California, Berkeley. And I understand today she's going to be talking to us about her the new YAP-TEAD inhibitors from Vivace. Thank you so much.
First, thank you for your invitations to join this discussion today. I feel really honored to be able to participate here. And so today I will tell you about our clinical candidate, VT3989. So first introduction to Vivace Therapeutics. Vivace was founded by Quinliang Guang and Shen Ding back in 2015. And the company's mission at the time was to target the Hippo pathway to discover and develop novel drugs. And what's unique about our company is that it's completely virtual. So unlike most biotech companies or pharma, we have absolutely no lab in our company. So everything is done by contract research organizations around around the world, and mostly located in China. But we also have collaboration with academic investigators, and as of today, we have more than 80 collaborators globally, because the CROs, they can run assays. As you have heard, Youchen is a CRO that can run amazing kinase assays. But most of these CROs, they do not have very unique models or very complicated models for experiments, like you have heard already, fibrosis models, cancer model, orthotopic models. So we do collaborate with academic investigators like yourselves that have these models that are much more complicated, and we provide tool compounds, and this is how we generate many of our data. And right now, we have our clinical candidate in trials, and the first patient enrolled that was dosed with our T inhibitor was in March 2021. And right now, the clinical trials are still ongoing, and so I'll share with you some of that data today. First, how we started screening for compounds that target the Hippo-YAP pathway. So as you know, the Hippo-YAP pathway, initially, it was very undruggable because there's so many signals that feed into the pathways, as Lilian has presented. And the NUAK, for example, the upstream kinases have many groups like yourselves are targeting to affect the YAP localization. So for us, when we started, we decided to do a phenotypic screening using the YAP reporter system, as illustrated here by this diagram. And we screened over 160,000 compounds, and we were able to identify several screening hits. Illustrated here are two of them, VT101, VT102. The IC50 of these compounds were extremely high in the reporter assay, about 400 nanomolar. But we also check how they affect the YAP phosphorylation or localization. We actually saw no effect at all, unlike the NUAK compound. These compounds do not affect YAP phosphorylation or nuclear localization, but we found that they actually affect YAP-T protein-protein interaction. So we took these compounds and started to do structural analysis, optimizing the potencies and also optimizing the drug-like property of these compounds. And we also later discovered that these compounds block the palmitylation of T protein in cells, as well as in purified protein assays. So a little bit about these compounds. So like I said, 101, 102 are the screening hits. And after our SAR efforts by our medicinal chemists, we have now optimized these compounds with good potency. So going from 400, 500 nanomolars to single-digit nanomolar for VT103, for example, 10 nanomolar for 104. And we also have really enhanced their PK property. So these compounds have extremely long half-lives in mice, 13, 24 hours half-life. They also have good oral availability, as you can see here, 75, 78%. And couple of compounds I want to mention is that these VT106, VT107. 107 is very potent, about 5 nanomolar in the YAP reporter assay. The half-life isn't as great, but this pair of compounds are excellent for in vitro studies because 106 is a mirror image of 107, and you can see it's much less potent. So this pair of compounds offers great tools for in vitro studies. And for our and then we determined that our compounds can bind to T protein directly, and is binding to the palmitoylation pocket of T. So a little bit of background here. It has been published by many groups that T transcription factor in the central pocket. There's a central pocket, and there's a conserved cysteine that's palmitoylated on the T transcription factor, and the palmitate sits in the central pocket. And it turns out our small molecules can occupy the central pocket as shown by this crystallography here, 105, which is another T compound that we have made. And so you can see the cysteine in the crystal structure is located here, this conserved cysteine. There's a lysine here, and this small molecule sits nicely in the central pocket. And our clinical candidate 3989 also binds similarly in the central pocket. And so some data here, what we found that 3989, we found that it can selectively cause anti-cell proliferation effect in NF2 deficient or NF2 mutated cell lines. So NF2 is a gene encoding Merlin, which is upstream of the Hippo core complex. It negatively, positively regulates the LAT. So when it's deleted, YAP-TAZ becomes hyperactivated. And we found that our T palmitoylation inhibitors selectively inhibit proliferation of NF2 deleted mesothelioma cell lines. And you can see on the other hand, the compound has no effect on cell lines that have wild type NF2. All of these lines here are mesothelioma cell lines. And by thermal shift assay using recombinant protein, we also found that 3989 interacts with T1, 2, and 3, as well as T4. And using palmitoylation assays in cell assay, we were able to see that 3989 can block palmitoylation of T proteins in cells in a dose-dependent manner. And in vivo, we show that 3989 can also inhibit the tumor growth of mesothelioma in mice, and with a minimal dose of 3 mg/kg daily dosing. Okay, so that was preclinical data. So we, as I said, our first patient that was dosed with 3989 was back in 2021, and the goal of the phase 1 trial was to determine maximum tolerated dose. And so on the left side, you can see that this is a dose escalation. And then we went as high as 200 milligram daily. However, we did not find any dose limiting toxicity. And on the other hand of this diagram, we're trying also intermittent dosing of the compound, starting with 200 milligram, 2 weeks on, 1 week off. And so we're in our trials right now, we're looking for the most optimal dose and also a dosing schedule. And the only toxicity that we've seen was proteinuria, because there's an on-target effect on podocytes in the kidney by T inhibition. And we also seen, besides evaluating safety of our first-in-class, first-in-human compound, we also wanted to see if there's any efficacy. So we enrolled mesothelioma patients as well as other NF2 solid tumors. And as you can see in this table, we have seen anti-tumor activities in many patients. For example, this patient here that have the dual pleural and peritoneal mesothelioma received the inhibitors at 200 milligrams, 2 weeks on, 2 weeks off, had actually minus 81% response, and this patient is still on trial. We also saw many other patients with response as well. And this is just a waterfall plot showing only the mesothelioma patients. So you can see many patients had their tumors shrinkage induced by after the 3989 treatment. Just showing you two cases to here. So this is what I mentioned, the 22-year-old female patient. This patient had no detectable NF2 mutation, however, but she was able to have her tumor shrink with 3989. As you can see in the scan here, there's this tumor and tumor shrinkage, and this patient is still on treatment. This suggests that the compound, the drug is very durable and also tolerable. We also have another case here, 51-year-old male patient with advanced peritoneal. Also, this patient did not have detectable NF2 mutations, but you can see the tumor shrank after 3989 treatment. So in summary, 3989 is shrinking the tumors, and the toxicities of albuminuria due to on-target effect on podocytes in kidney. Many of patients are still on study, suggesting that the responses are quite durable. And also I should mention the toxicity of albuminuria is reversible. So when the patient went on intermittent dosing, their albuminuria is reversible and would drop down to normal range. And so besides single agent activity, we're also considering combination strategies for our 3989. And that is because in the literatures, we have seen many reports that YAP-TAZ activation could be responsible for the emergence of drug-tolerant persister cells or dormant cancer cells, and this leads to the drug resistance. So we started combining 3989 with other targeted therapies, in this case, osimertinib, which is a EGFR inhibitor. So you could see that osimertinib single agent 2.5 mg per kg daily dosing, the tumors actually relapse on treatment. When we added 10 mg per kg 3989 on top, you could see this relapse is delayed. And then with 30 mg per kg 3989, during the duration of the study, almost 80 days, we did not see any tumor regrowth. And we also tested this in a PDX model of lung cancer with the exon 19 deletion. And you can see in the combination effect by adding 3989 on top of osimertinib, we saw reduced tumor relapse as well as delayed tumor relapse. Using another EGFR inhibitor, lazertinib, we saw the same effect. So this is really indicating that blocking YAP-TAZ activation during treatments by targeted therapies such as EGFR inhibitor, using our 3989 compound, we can enhance the durability and also efficacy of the targeted agent. And besides EGFR, we also looked at many targeted agents in this TKI pathway, such as MET inhibitor, BRAF inhibitor, MEK inhibitor, everolimus, and I'll just showing examples here, different EGFR inhibitors again, and here's a MET inhibitor, MEK inhibitors, as well as the KRAS G12C inhibitor. And you can see in all cases, combination with 3989 with the targeted therapy, you can see a very strong tumor combination inhibition combination effect. There's delayed tumor regrowth and also enhanced efficacy. So in conclusion, what I've shown you today is that our clinical candidate 3989 shows a good safety profile in the clinic and also has shown anti-tumor activity. I've also talked to you about combination strategy. So right now, we are also planning to combine V989 with some other targeted agents, and we'll be starting the clinical trials sometime later this year. And the strongest message here is that the Hippo pathway, unlike what people have thought maybe 8-10 years ago, the Hippo pathway is actually now very druggable, as you can see with NUAK and also with the T inhibitor. And then the Hippo pathway is also now a validated target for cancer therapy, as we now have T inhibitors in the clinic showing activity. And finally, is our company, although is virtual, we have some few employees, actually very small company that are full-time, but we work with many other scientists and collaborators in the world. And thank you for your attention.
Thank you so much for a great talk.
I guess, Liliana, I think the next stage is a 1-minute takeaway from each of the speakers. I think we can start with Catherine.
Okay. Thank you.
Sure. So the takeaway message from my talk is that NUAKs are frequently amplified in various cancers. And then we have identified a positive feed-forward loop in both the cancer context as well as in the fibrosis context. So in the cancer, it's NUAK2 works together with YAP-TAZ. So YAP-TAZ induce NUAK2 expression, which in turn inhibits LADs to enforce a strong YAP-TAZ transcriptional program, which drives tumorigenesis. And in fibrosis model, TGF-beta and YAP-TAZ can induce NUAK1 expression, which in turn activates YAP and SMAD signaling, which in the end drives fibrogenesis.
Thank you, Catherine. Methvin? Methvin, you're on mute. I think Methvin can't hear us. Methvin, I think you're on mute.
Methvin, you're on mute.
So, yeah. Oh, sorry. How is that now?
Yeah, that's good.
Thanks, you're
Thanks.
muted.
Yeah. Can you hear me now?
Yeah, we can hear you loud and clear. Yeah.
Okay. Yeah. No, I was saying it's quite exciting to see a lot of times, your genetic biology don't always phenocopy small molecules. I mean, there are a few cases sometimes where, you see amazing CRISPR knockout and stuff, and then, you can't mimic a lot of that with your small molecule. But it looks like now this is a case where one, you had a pathway, which historically was thought to be undruggable, and now, you genetically validate it with small molecules. You actually have, other mechanisms in the pathway, the TEADs. And now, we have a new angle, NUAK inhibitors, which is really phenocopying what we see genetically. So to me, that's really, really exciting to see those things lining up. And so there's a lot of promise to really jogging this pathway. And it's also exciting to see some of the, chemo resistance and some of those combination data. So I think, it's for me, it's a very, very exciting and hot area.
Thank you, Methvin. Tracy?
Yes. So to me, I think it's really interesting that a transcription factor would be palmitoylated. Usually you think of palmitylation modification, fatty acid modification on protein as being on the surface of the protein and allowing the protein to be secreted to cell membrane or outside of the cells. But seeing a transcription factor such as TEAD being palmitoylated, and then on top of that, this palmitylation site where the palmitate sits is inside the protein in a central pocket instead of on the surface of the protein. So for me, that's really exciting. So one of the questions that we can talk about would be why a transcription factor would be palmitoylated and what's the purpose of this? And I think so far, T transcription factor is the only transcription factor that's known to be palmitoylated in such a way. And then now with that being said, with a central pocket in the protein, it becomes easy, quote unquote. I can't speak for medicinal chemists, but now you have a central hydrophobic pocket. It really makes it easy to target this transcription factor, like you would be able to target kinase or other proteins that has pocket for small molecules to bind and exert activity on the molecule. So with that, we were now able to see many T palmitylation inhibitors coming out from multiple companies and also academic groups. And Vivace, we were lucky that we were able to have a compound now being tested in the clinic and as a first in-patient molecule. And we're excited to see that the safety profile is pretty good. Proteinuria, albumin urea are the ones that we're seeing now with different dose as we escalate the doses, and we're trying different intermittent dosing schedule, and really shows that the Hippo pathway is druggable now, and also it's validated target for cancer therapy. So, and we're excited to see more T inhibitors coming online and perhaps would do better than 3989, but it's very exciting that now we can offer patients that have misregulated Hippo-YAP pathway with a therapy. And also later on, we can also target these drug-resistant, drug-tolerated persistent cells in combination strategies.